So the advent in 1977 of molecular phylogeny recast and restructured fundamentally how we look at the emergence of life on planet Earth. Now, an important context for our modern day understanding of the emergence of life is to look at how different and how drastically changing and evolving itself the whole concept of evolution has been over the last 150 years, and in fact even deeper into human history than that. So what we want to do is take a look at the changes in evolutionary thought and how people had tried to graphically structure the tree of life, and then see how fundamentally different from that than the departure is for the results of Carl Woese and his colleagues here at the University of Illinois. So we need to start all the way back actually in 1674 when Antony van Leeuwenhoek, who was a scientist in Holland, developed the first microscope. And that first microscope was actually a brass plate with a few very crude lenses and a setting screw that allows you to change the angle and therefore the magnification of the lenses. Well, Leeuwenhoek is very famous because of that development of the microscope, and he's the first one who actually published an observation that there was life on a very small scale. And that microscope was strong enough for him to see 1 micron cells, some of which were bacteria, some of them were eukarya, the more advanced forms of cellular life on the planet. And that publication in the Proceedings of the Royal Academy of Science was a benchmark. It was the first time that human beings had seen life active at that kind of scale. The next step in the process was about 100 years later, and in 1735, Linnaeus, who was a Swedish scientist, decided to use primarily the observations of the diversity of life on planet Earth from morphology, the size, the shape, the color, some of the attributes of how the organisms lived, try to use those to break all of life into pieces and compartments that could be used to look at the structure and eventually the historical evolution of life. And Linnaeus came up with 2 kingdoms, he called them. One were the animals and one were the plants. And so Linnaeus was critically important for developing the ability to describe morphological differences and move forward with that kind of what we call taxonomy, that descriptive taxonomy of what was living. Well, and again, about another 100 years plus had passed, and in 1866, Haeckel decided to start structuring not only morphologies you could see with the eye, but also some morphologies you could see under the microscope. Now remember, this is long, long before the genetic tools were available. So this was using whatever tools of observation were possible at these times. And what Haeckel put together was a very different kind of tree. Again, Linnaeus had animals and plants. Haeckel, as you can see from this, also had plants and had animals, but also had another small group of organisms called the protists. And these protists were single cells or sometimes multiple cells, but they were small and they moved around, and you could see they had these different morphological features, they had some different colorations. But then also he broke things into another group called the Monera. And the Monera were what we call in the present day these single-celled microbes that eventually were bacteria and archaea. So the Monera was kind of a loose grouping of single-celled organisms. The protists were the more complex yet still small organisms, and he kept with the plants and the animals. Well, another 100 years plus passed, and there was a benchmark work by a scientist named Whittaker in 1969. And you can see from the Whittaker tree of life, there was this concept of having eukaryotes, and that was one of the earlier times that eukaryotes had been coined, and then we had prokaryotes, and those were all the single-celled small organisms. So going from prokaryotes to eukaryotes, and you can see that tree makes it a very linear progression of the two, implying that eukaryotes evolved directly from the prokaryotes and all the prokaryotes. And you can see some of the same names were kept. The Monera were part of that prokaryote group, and the protists, again, these more complex small single cells. So the Whittaker tree is actually what is still used in a lot of applications within science today and can still be found in some textbooks. Well, the advent of the 1977 publication by Woese and Fox, and then the ensuing publications, especially Woese et al. 1990, reset fundamentally the stage of how we understand that the structure of the evolution of life on this planet took place. So all these came together to produce a drastically different looking tree of life. And in this tree of life, you can see that we're hovering in a helicopter, if you will, looking down at the uppermost branches of that tree of life. And in the middle, we have an origin, we have a root or a stalk, versus having these three branches, bacteria, archaea, and eukarya, coming up and out. The importance is that the context of the history of the evolution of the idea of a tree of life itself really sets the stage for us, because it compares and contrasts what was thought of as being simpler life and more complex life, and what came first and what came second. Instead of some of those paradigms and hypotheses that were put up, and very reasonably based on morphology, the genetic tool, the molecular phylogeny, allows us to quantify these relationships and look deep into time to see how diversity on this planet is actually structured in terms of its capabilities of lifestyles, ecology, but then even more importantly, its genetic composition.